Okay, we'll continue. All right, good morning everyone. Please confirm me, am I audible to everyone? All right. Good morning everyone. Fine. So in the yesterday class, we have started studying about the cyclic photophosphorylation as well as the non-cyclic photophosphorylation. I hope everyone remember, in case of a cyclic photophosphorylation, there is only involvement of one photosystem, and I want every one of you to compulsory remember that here there is only production of the ATP, whereas in case of a non-cyclic photophosphorylation, most importantly there is an involvement of two photosystems. And always remember, from the P680 who is accepting the electron first? Phyophytin, and from the P700 always electron is accepted by the A0. Okay, A0 is also called as modified chlorophyll A, and A1 is said to be what? Phylloquinone. No point of getting confused over here. Okay, we understand this very clearly. Now I hope you all remember, in case of a non-cyclic photophosphorylation, in the yesterday class we understand how we produced NADPH. But it was very clear that in case of non-cyclic photophosphorylation, we also produce the major energy equivalent, that is the production of ATP. We will understand how exactly we are going to produce the ATP in the non-cyclic photophosphorylation in a elaborative way. Now, the most important point I want you people to remember here is that, good morning, continue everyone. The synthesis of ATP can be mediated by a special force which we generally call it as PMF, which is also called as proton motive force. By this only we can synthesize the ATP very easily. Okay, tell me what are the major cofactors that are required for the photolysis of the water apart from the manganese? So we require the chlorine as well as we require the divalent ion calcium also. Now listen, first understand the concept very clearly, then you will get a clear cut idea. Okay, just imagine a condition. Listen, as I said that ATP can be synthesized by a proton motive force, which means that initially the concentration of the protons are higher in the stroma, the concentration of the protons are lower in the lumen initially. Now all of you pay attention. You can see here, this is the thylakoid membrane or the lamellae, this is the photosystem 2, and this will be the photosystem 1. This is called as F0 F1 complex, also called as ATP synthase enzyme. Then plastoquinone, cytochrome B6F, these are all what? Several electron acceptors. Now all of you try to understand the concept correctly, just listen. So from the P680, who is the first electron acceptor? Phyophytin, undoubtedly. Then everyone please continue. From the phyophytin, electron moves to where? Plastoquinone. Now understand very clearly. Plastoquinone, upon accepting the electron, it is readily reduced to the PQH2. Upon accepting the electron, it is readily reduced to the PQH2. But the point is that if it wants to reduce it to PQH2 along with the electron, it compulsory requires the proton also. Now from where the proton is coming from? It is coming from the stroma. Upon accepting the proton from the stroma, electron from the phyophytin, plastoquinone reduced to what? PQH2. Now the question is very clear, PQH2 never keep electron with it. It has to pass to the next electron acceptor complex. Then PQH2 passing the electron to whom? Cytochrome B6F. Upon passing the electron, PQH2 again oxidized back to PQ. And what about the proton? Proton, it is going to release into the lumen. Okay, once again every one of you try to understand correctly. Initially, whenever the pheopoietin passed the electron to the plastoquinone readily reduced to the PQH2, and in order to get reduced to the PQH2, it also accepts the proton from the stroma. Then PQH2, it again send the electron to the cytochrome B6F and it is oxidized back to the PQ, and that proton is released into the lumen. Now what is happening? All the protons are coming from stroma to lumen. At the same time, in the yesterday class we read, whenever the water molecule break down, what do we got? We got 2 proton, we got 2 electron, and we got half oxygen. This is the oxygen we used in the respiration. And what happens to electron? Electron is taken by the PS2. And what happened to this proton? Again the protons are released into the lumen only. Obviously released into the lumen. Then what is happening? The concentration of proton will be higher in the lumen compared to the stroma. Then again all the protons, to maintain the charge balance, from the lumen they are start moving towards the stroma. How they will move? They starts moving via a specialized complex. We call it as a F0 F1 complex, also called it as a ATP synthase enzyme. Now what is the function of proton here? Always remember, whenever the proton is moving, it moves in the form of a wave like this. So whenever it is moving in the form of a wave, it generates energy. That energy can be used by this F1 particle, combine the ADP with PI, hence there is a formation of ATP. So that's why we can say that the ATP production is exclusively mediated by what force? Proton motive force. No point of getting confused over there. So protons are moving from the lumen towards the stroma, which generates energy. That energy can be used to join ADP with PI, hence there is a formation of ATP. So this is only happening where? This is only happening in the non-cyclic photophosphorylation only. Now let's try to understand very correctly what are the major differences between the cyclic and non-cyclic. Anyway, we know that in the cyclic only one photosystem is involved. Here both photosystems are involved. Most importantly, here P700 is the reaction center. Here P680 is the reaction center. The most important one, here there is no photolysis of water is taking place. Correct na? Here 100% photolysis of water takes place. Wherever the photolysis is taking place, undoubtedly oxygen is going to be released over there. Wherever it is not happening, oxygen never be released. In the cyclic we studied both energy equivalent, only energy equivalent produced, reducing equivalent never be produced. But here both energy equivalent, reducing equivalent, both can be produced. And they won't ask this much simple question. Mostly the questions comes from what we can say inhibitors. The inhibitors that blocking both cyclic and non-cyclic. This is very, very important. You all people has to remember compulsory for the exam. So there are a lot of herbicides. They actively block the electron flow. Among those, the major ones are DCMU, triazine, as well as the paraquat. It is very important. You people has to remember almost all the names. Now listen, this is the DCMU. No need to remember the structures, which is also called as dichlorophenyl dimethyl urea. Next one, paraquat, also called as methyl vialogen. No point of getting confused over there. Now all of you understand the concept very correctly without having confusion. P680, see this, everyone focus. This is the area how the electrons are flowing from the P680 to the P700. Now this flow can be blocked by the DCMU. And where exactly it is blocking? Where exactly it is blocking? It is exactly blocking in between QA to QB. So exact, see, in a simple words, DCMU is generally blocking the electron flow between P680 to P700. But the point is very clear, exactly where it is blocking is between the QA and QB. So see, anywhere we block it, obviously the entire electron flow will be disturbed automatically. Then if you look into the paraquat is actually blocking the electron flow that is involved in the production of NADPH. I hope it starts from where? A0, and finally who is the electron acceptor? NADP plus. This paraquat can block the electron flow in any way, or whether it is between A0 to A1 or iron sulfur X at any point, but it is generally blocking the electron flow that is involved in the production of NADPH. No point of confusing over there. It's very much important to remember maximum amount of inhibitors. Okay. Slowly think, don't be confused, and answer this 76 question. Everyone online people answer this question number 76. Slow. At least it's very important to spend 30 to 45 seconds on the question, even though we know it. Then before R, okay, others? Yes. All right, look into this. So we generally know that both DCMU and paraquat block the electron flow in photosystem 2. Wrong. Both cannot block one single photosystem. Both DCMU and paraquat block the electron flow in the photosystem 1. That is also wrong. DCMU block which one? Photosystem 2. Correct na? Paraquat blocks the photosystem 1. So option 3 is also a wrong option. Then DCMU blocks the electron flow in photosystem 2, whereas paraquat block in the photosystem 1. Hence the correct answer will be what? S. Right. So that is what the difference. Correct. That is the difference between the cyclic and non-cyclic photophosphorylation. Now, before moving to the dark reactions, it is very important we have to read some other important mechanisms. Among those, it is very important to understand the physical separation. What exactly this physical separation, and what is the significance of this particular physical separation actually? All of you understand this very correctly. Just 1 second. Just a minute. One second. All right, everyone of you focus, what is this exactly physical separation of the two photosystem? Have you ever thought why electrons are not flowing directly between PS to PS1? Why it is not directly flowing? Why always electron flows via several electron acceptors? It is moving via pheophytin, plastoquinone, different electron acceptor. Why the electrons cannot flow directly between the two photosystems? If the electron directly flows between the two photosystems, you cannot produce energy equivalent, you cannot produce the reducing equivalent also. Compulsory there is a requirement of electron carriers between the photosystems. Now for that reason, it is very important that we have to separate the two photosystems that are located on the thylakoid membrane. Now all of you observe the picture very correctly. Everyone focus. This is said to be the thylakoid membrane, and of course the matrix which is called stroma, and inside the fluid filled structure which we generally call it as the lumen. There is no point of getting confused over there. Now every one of you understand very correctly what all these are all generally LHCs, okay, or you can say cluster of thylakoids also you can call. Now all of you focus on the picture very correctly. This is light green color, dark green color one PS2, light green color one PS1, ATP synthase enzyme, the blue color one is the cytochrome B6F dimer. Now observe the picture very correctly. Where exactly the PS2 is located? Location is very important. You can see that PS2 only located between tightly oppressed thylakoids. What does it mean by the word tightly oppressed or stacked of thylakoids? Between the thylakoids it got merged. It is not freely arranged. That's why we can call it is arranged between the tightly oppressed thylakoids or stacked regions of the thylakoid. Now all of you focus on the picture. This is the PS1 and this is what? ATP synthase enzyme, where they are located between the non-oppressed thylakoids. Non-oppressed means where there is no cluster of the thylakoids. That are unstaked regions we will call sometimes. And what about the cytochrome B6F? It is present on the tightly oppressed also or non-oppressed also. So what we can say that cytochrome B6F is evenly distributed. Cytochrome B6F evenly distributed. PS2 located between the unstaked regions or the what we can say, staked regions are tightly oppressed thylakoids. PS1 and ATP synthase is located on the unstaked region or loosely oppressed thylakoids. This is how actually they will be arranged. Everyone quickly answer this question. Very quick. Good morning everyone, please continue. Sorry, this one. Everyone online? Option 4. Relax. 2. What's the answer? Nihila, what is the C? What happened? Look into this. About every one of you, what is the answer? Online people. All of you look into the given question. Photosystem 2 is located predominantly in the staked regions, which is obviously true statement. It is located on the tightly oppressed thylakoids, which we generally call as staked regions. Right statement. Photosystem 1 is found in the unstaked. That is also right. Along with the photosystem, ATP synthase also located where? Where it is located? That is also on the unstaked. D is also right. Cytochrome B6F is evenly distributed, but they said that it is confined to only stroma, which is a wrong statement. Hence the correct answer will be the option 2. Is it clear everyone? Online people, correct. Good morning. Answer is 2. Clear, understand? These are all what? Various type of a light reactions. Okay. Now Once we read the dark reactions, we will come back to the photoprotective mechanisms in an elaborative manner. Just, I think I kept it there only, yeah. Alright, so now let's understand the most difficult part of the photosynthesis, that is called as dark reactions, right? I hope, correct. I hope every one of you remember my words very clearly, that dark reactions are the area, or dark reaction are the reactions where final fixation of the carbon into glucose takes place. Correct. I hope you all remember, dark reactions doesn't mean that they are occurring in the dark, which means that they are light independent reactions. Whether the light is present or light is absent doesn't matter, they always takes place. Now the point is very clear, it is also called as because we are fixing the carbon into glucose. That's why we can call carbon fixation. At the same time, we are assimilating the carbon. What does it mean? Plants assimilate, incorporate the carbon to use it into sugars. That's why we also call it as carbon assimilation reactions. No point of getting confused over there. Now next, this is only most important. I want all of you to focus on it very correctly. Based on the dark reactions, now where this dark reactions actually takes place? In the stroma. What is the major reason it is taking place in the stroma? One of the major reason is that all the enzymes are located over there. What is the major enzymes? Rubisco, followed by what is the other enzyme? What the enzyme used by the C4 and CAM plants? PEP carboxylase. They are all present in the stroma only. So now you see this, whatever the ATP and NADPH that we are produced in the light reactions, what we are doing? We are using them, with the help of by using this energy, plants are reducing the carbon into glucose. Hence, at this particular condition, we do not require light. What do we need is only energy, right? So now, based on the carbon fixation reactions, all the students please try to understand my point. Based on the carbon fixation reactions, we categorized all the plants on this earth into C3, C4, then followed by the CAM plants. No doubt about it. And how did we categorize them? That is also a very important point to be understood. 95 to 97.5% of the plants on this earth belongs to the C3 plants. Now, what about the C4 plants? Listen my statements correctly. C4 plants are those type of plants which are living under high light intensities. I am not at all using the word desert, okay? I am just saying that high light intensities or a little high temperature. In more simple words, I can say to all of you, more than 18,000 types of angiosperm families will follow the C4 pathway. More than 18,000 types. Then what about the CAM plants? The plants which are belongs to a family called as Crassulaceae family. In simple words, you can call it desert plants. What are the example? Agave, succulents, euphorbia, pineapple, bromeliads. We're going to read it. So, based on what we categorize, based on the fixation of the carbon, we categorize them into three major types. Now, let's try to understand one by one more elaboratively. It is simple to understand, but it makes you confused if you don't understand. You read the C3 cycle multiple times, but C3 cycle always make you confused. Now first let's understand C3 cycle, which is proposed by Melvin Calvin and his team, which they awarded Nobel Prize. We also called as C3 pathway. Why do we call C3 pathway? The stable or intermediate product that is produced is the 3 carbon. That's why we called as C3 pathway. Now, the C3 pathway generally happens in 3 steps. One is called fixation, second one is called reduction, and the third one we generally call it as the regeneration. I'll explain everything elaboratively, just focus. fixation of the carbon. What is our ultimate goal? Fixing the carbon into glucose. Point is very clear, we can't fix the carbon directly into glucose. So first we will fix the carbon into what? 3 carbon containing compound. What is the 3 carbon containing compound? 3-phosphoglycerate. That's why we can call C3 pathway. Now that 3 carbon containing compound, which is called 3-phosphoglycerate, we are reduction into glyceraldehyde 3-phosphate. This is also 3 carbon, this is also 3 carbon. We just reduced it. Then finally we are regenerating something called as RUBP. I'll explain step by step elaboratively, just pay attention. Okay. Meanwhile, answer this question all the students quickly. This is the question appeared in the 2023 December afternoon shift. This much fast, trust me Sheetal, I got it, I am just saying I want you to be more patient. Why? Because sometimes we'll tend to choose the wrong answers. Alright, this can be easily answered as she said. Electrons from P680 first move to what? Quinone, then to pheophytin. Absolutely wrong statement, because first to pheophytin, then to the quinone. So based on the options also we can come to a conclusion that this is BCD. Anyway, let's see that P700 received the electron from plastocyanin. PC is the electron donor for P700. NADPH is produced at the end of light reaction. Of course, ATP also produced at the end of the light reactions. The hydrogen ions produced during the light reaction get concentrated in the lumen. Again, they move from lumen to the stroma. By that, they'll synthesize the ATP molecules. Correct. Okay. Anyway, focus everyone. First, listen to me very carefully. First, let me completely explain it. Later only you answer all my questions. Okay. So, first listen to me carefully. That's all. Okay. All of you pay attention. It is just all about understanding the concept. What is our ultimate goal of a Calvin cycle? Ultimate goal of the Calvin cycle is fixing the carbon into glucose. That is ultimate goal. Listen. And if we want to fix the carbon into glucose, someone has to accept the carbon. Who is going to accept the carbon? Who is the acceptor of the carbon? The acceptor of the carbon is said to be the RUBP. Ribulose 1,5-bisphosphate. What's that? Ribulose 1,5-bisphosphate, which is the acceptor of the carbon. Now tell me how many carbons RUBP is? 1, 2, 3, 4, 5 carbon. Correct na? It accepted 1 carbon from the atmosphere. 5 plus 1, 6 carbon. And I hope you understand. 5 plus 1, 6 carbon, that is completely unstable compound. Completely unstable. So I want to make it stable. And we have a rule that our intermediate product should be how many carbon? 3 carbon. So I am having 6 carbons in hand. How many 3 carbons I can produce? 2 molecules of 3 carbon I can produce. This, there is no confusion at all. Everyone got a clear-cut idea. So now follow my words very correctly. Just listen what exactly I am explaining to all of you. Exactly. Understand my words. Focus all of you. In order to produce half glucose, that's all. In order to produce half glucose, Calvin cycle has to turn how many times? 3 times. In order to produce 1 glucose, Calvin cycle has to turn how many times? 6 times. No confusions. Which means for 1 turn, 1 RUBP will come. Let's calculate for half first. 1 turn, 1 RUBP will come. So 3 turns, 3 RUBP will come. 1 RUBP is how many carbon? 5 carbon. 3 into 5, 15 carbon. Very good. As we said, every time 1 RUBP accept 1 carbon. So 3 RUBP accept 3 carbon. 15 plus 3, 18 carbon. And what is our rule? Our stable compound should be how many carbon? 3. So how many stable compounds we will get it? 6. 18 by 3 will get the 6 molecules of the 3-PCA. See that everyone? In order to produce half glucose, cycle is turning 3 times. So 3 molecules of RUBP combines with 3 molecule of carbon dioxide, 15 plus 3, 18, and it is going to give rise to what? 6 molecules of 3-PGA, right? Since our stable product is the 3 carbon, that's why we can call what cycle? C3 cycle. No point of getting confused, just think properly and understand. So someone has to catalyze this reaction. What is that enzyme? Catalyze this reaction is Rubisco. So always remember, most of the people will get confused between the Rubisco and RUBP. RUBP is a carbon acceptor. Rubisco is the enzyme. You are getting the difference. Alright, we produced what now? 6 molecules of 3-PCA. What is this step is called as? Fixation. Very clear. Now understand, out of 6 molecules of 3-PCA, all these 6 molecules of 3-PCA, listen, second step, which is called reduction. They are getting reduced into what? 6 molecules of what? G3P, glyceraldehyde 3-phosphate. 3-PCA is, each 3-PCA is how many carbon? 3 carbon. Each G3P is how many carbon? 3 carbon. So there is no change in the carbon number. What has happened? Just reduction happened. You got the point? Now that is very clear. So reduction is not that much easy process. It requires a lot of energy. In order to reduce one 3-PCA into one G3P, we require one ATP and one NADH. Hence we have 6 molecules of 3-PCA. We require 6 ATP and 6 NADH. I hope it's clear for everyone. Now out of 6 molecules of G3P, this is the crucial step. Only how many molecule of G3P is coming out of the cycle? You can see that only 1 molecule of G3P is coming out. Only 1. And giving rise to what? How many glucose? Half molecule of glucose. Clear? Now tell me how many G3P are left? 5. 5 means how many carbon? 15. Correct na? Each one is how many carbon? 3 carbon. Then use the brain. By using 15 carbons or by using 5 molecule of G3P, how many RUBP I can regenerate? Obviously I can regenerate 3, since RUBP is the 5 carbon containing compound. This is called what? Regeneration. Correct na? In this regeneration, that is, while we are regenerating the G3P into RUBP also, we use how many ATP? 3 molecules of ATP. Clear? No confusion here. Understand? So I'll summarize once again, then you people answer my questions slowly. Everyone answer the question that what I will give. Just think properly first. I'll summarize. In order to produce half glucose, cycle is going to turn 3 times. For 1 glucose, 6 times. Very clear. So who is the carbon acceptor? RUBP. RUBP accept the carbon and convert into, so 3 molecule of RUBP accept 3 carbon and giving rise to 6 molecules of 3-PCA. Now 6 molecule of 3-PCA getting reduced to the 6 molecule of G3P by spending 6 molecules of ATP and 6 molecules of NADPH. Out of 6 molecule of 3-PCA, sorry, G3P, 1 is coming out and producing half glucose. Then remaining 5 molecule of G3P are regenerating into 3 molecules of RUBP by spending 3 molecules of ATP. Clear? Every one of you clear? I hope all the students are very clear. Now I am requesting, use your brain. Now you, whatever the answers you want to give, spontaneous, speed, slow, but think and give. How many number of carbon dioxide, how many number of ATP, how many number of NADPH are required to produce, simple question I am asking, 2 molecules of glucose and 1 molecule of sucrose? Consider this is the question you will get in your December exam, right? Calculate it. Every one of you please answer. How many number of carbon dioxide, ATP NADPH are required to produce 2 molecules of glucose and 1 molecule of sucrose. Calculate dear, calculate. Everyone. Done. Tell.
For 2 molecular glucose, 12 CO2, 36 ATP, 24 NADH.
Okay. Next for sucrose.
Glucose plus glucose.
मुझे नहीं पता है. Sucrose, glucose plus fructose. Same.
For sucrose, 24 CO2, 72 ATP, 48 NADH.
Okay. Others? Okay, all right. Always it is idealized to calculate for 1. When you calculate for 1, it becomes so easy. So let's calculate for 1 glucose molecule. Tell me, for 1 glucose molecule, cycle has to turn how many times? For half glucose, cycle has to turn how many times? 3 times. For 1 glucose, 6 times, which means we require 6 carbon dioxide. I have asked first 2. 6 into 2, 12 carbon dioxide. In order to produce half glucose, how many ATP required? 6 plus 3, 9. So in order to produce 1 glucose, how many we required? ATP. For 1 glucose, how many we required? 18. Correct or not? 18 ATP for 1 glucose. Correct na? If it is 2 glucose, 36 ATP. Correct or not? Then to produce half glucose, how many NADH required? 6. For 1 glucose, 12. So for 2 glucose, 24. Correct na? This is for how many? 2 molecules. And 1 sucrose equal to 1 glucose plus fructose. Anyway, fructose is actually later converted into the glucose only. Then how many? 1 sucrose means 2 glucoses. How many? It will be same or different? Obviously it is same only, right? That is what I asked. I asked for only 1 sucrose molecule. Hence obviously 12 carbon dioxide, 36 ATP and 12 NADH are required. Correct or not? Is it clear everyone? Online people understand clearly? Every one of you please confirm, is it clear online people? Okay. There is a very simple question. How many number of G3P are required to produce 20 molecules of glucose? I hope it's clear for everyone online people. Now tell me, how many number of G3P are required to produce 20 molecules of glucose? Anyone having any kind of a doubt, you can ask. Parul, you got it correctly? You understand it? How many kauna? How many? 120. How many number of G3P are required for 20 molecules of glucose? 240. Oh my God. What is the question I asked? What is the question I asked? How many G3P are required to produce how many glucose? How many? What is the answer? 240. All right. Look into this. Listen. 1 molecule of G3P is giving rise to how much? How many glucose? Half glucose. In order to produce 1 glucose, how many G3P required? 2. Like G3P is how many carbon? 3 carbon. How many G3P are required to produce 1 glucose, which is 6 carbon? 2. That's all I asked. 20 into 2, how much? How it became 240? Right. 40 molecules of G3P are required. Correct na? Everyone understand this very clearly. Don't get confused. This is the question came in the NEET 2024 paper. Okay. Okay. Clear everyone? How many number of G3P are required to produce 20 molecules of glucose? No point of getting confused. Very, very good. Very good. Correct, Shivani. Okay. Last and final question. Think slowly, use your brain and answer my question. What is the net amount? What is the net amount of G3P that is involved in the production of glucose? That is the exact question. What is the net amount of the G3P that is involved in the production of glucose? 1. How 1? Because only 1 is coming out and producing half glucose. So how many used? 1. 1 is only coming out of the cycle. Correct, right? So that, what is the meaning of net here? How many are finally used? Not how many are generated. We generated 6, but used how many? 1. That is what exactly we call it as the net. I hope everyone understand that correctly. No point of getting confused over there. See that the question? Everyone please continue. No. I hope everyone got it correctly. What is the net synthesis of G3P on carboxylation of 3 molecules of RUBP in the Calvin cycle? What is the answer? 1 only. See, carboxylation of 3 molecules of RUBP, total how many G3P we got? 6 G3P. Correct or not? Out of 6 G3P, how many we used in the production of glucose? 1. That is called as what? Net synthesis. Correct now? So always you people remember for 1 glucose all the time. So for 1 glucose, 9 ATP we used in case of reduction step, 3 ATP we used in terms of regeneration step. Correct now? And how many NADH we used? 6 NADH. So always remember this. To make finally 1 molecule of glucose, we require 6 carbon dioxide, 18 ATP and 12 NADH into how many number they may ask. Okay. Every one of you please confirm me. Is it clear everyone till this particular slide? Confirm all the online people. Is it clear everyone? Don't get confused. It's a very simple question, but it is a little bit tricky question. So this is 2023 December part B question. Parul, try to answer this question. Huh? D. Okay. Photochemically generated ATP is consumed in which one of the following phases of the Calvin cycle? What is first of all ATP and anyway we are producing in the light reaction, so we only used in the reduction. We used the 6 here, and in the regeneration we use the 3. Correct na? So the simple one is the D. Is it clear everyone? Please let me know if anyone having any doubt. All right. Now again this is the question which is asked again in the 2023 December in a very different way. Finally, how many G3P are coming out to produce the glucose? 1. Correct na? 1. 1 give rise to how many glucose? Half. Simple. If 2 are coming, 1 glucose we get it. Now, you know very well, suppose if I am a plant body, I cannot transport glucose via phloem. Correct na? What I can transport? Sucrose, stachyose, ribose, raffinose, we will see that, arabinose, all these. Glucose cannot be transported. So undoubtedly glucose has to be converted into the sucrose, and at the same time glucose can also convert into the starch. Again the question asked in the exam, where glucose converted into the sucrose? Where is the site of site where it is converting into sucrose? That is cytosol. Why? Because there is an enzyme called as sucrose synthase enzyme. You got the point? Then same glucose is converted into a polysaccharide which is called as starch. What is the enzyme that forms the starch? Amylase. Right, that is present where? In the chloroplast. That is a very important question that is asked in the exam. Very good, correct. Now Now let's try to understand the most difficult step. No point of getting confused. Every one of you use your brain, properly answer the questions. Okay, this is all about how nicely you will understand. What is the enzyme here? Rubisco. The principal enzyme in the entire C3 cycle is the Rubisco. What is its function? Joining the RUBP with carbon and give rise to 3-PCA. But what is so special about this particular Rubisco enzyme? All of you try to understand my statements very correctly. There is no point of confusion. Use your brain and analyze it very correctly and tell me. So, Rubisco, the full form of Rubisco is ribulose 5-phosphate carboxylase oxygenase. This is the full form of Rubisco, which means Rubisco can fix the carbon also, Rubisco can fix the oxygen also. But what plant needed? Plant wants to fix only carbon. What is the point of fixing oxygen? There is no point. But since the enzyme is Rubisco, there is a possibility that Rubisco can fix the oxygen also. Now first question, why Rubisco is fixing oxygen instead of the carbon? What is that condition? There are multiple conditions. One of the major condition is high temperature. Right? Suppose in any metropolitan city you take a Chennai. Okay. Suppose the temperature is extremely higher. Then what is happening? Whatever the carbon dioxide that is available, that is completely solubilized at high temperature. Now what is left out? Oxygen. Oxygen also gets solubilized, but compared to the carbon dioxide, the oxygen is less solubilized. Then Rubisco is left over with what? Oxygen. Then obviously Rubisco is going to fix the oxygen only, which is a very dangerous or a waste reaction for us. Now again some students will get confused. This is also a statement that is asked in the exam. This is a Rubisco enzyme. Enzyme generally having what site? Active site. How many active sites are there in enzyme? Only one. So in the exam they will always say that Rubisco having a dual active sites for the carbon also, oxygen also. So most of the students will think that since it can fix the carbon and oxygen, the active sites are different. Active site is only one. Then what is happening? Carbon dioxide and oxygen is competing for that active site. You're getting the point? So whichever is more available, that will go and fix it. That can attach us to the active site, Rubisco will fix it. Simple point. Now what is our condition? Oxygen is higher. Why oxygen is higher? Because temperature is higher. As a result, soluble, because of that, entire carbon dioxide got solubilized. Now everyone don't get confused. Slowly think and answer the question. Anyway, the fixing of oxygen instead of the carbon by a Rubisco called as photorespiration. Is it useful reaction? No. It is extremely wasteful reaction. Now all of you understand. Who is the carbon acceptor in the C3 cycle? RUBP. How many carbon? 5 carbon. Plus there is 1 carbon dioxide in the atmosphere. Now this reaction is catalyzed by what? Rubisco. How many intermediates we will get? How many intermediates we will get? 3. How 3 dear? 5 plus 1 is 6. We will get how many? 2. Correct or not? 2 molecules of 3-PCA will get. Then only the Calvin cycle will be continued. Now what is the condition? Same RUBP. Now the temperature is higher. Is there any carbon now? What is available? Oxygen. Now use the brain. Now Rubisco fixed it. Correct now? Now slowly tell me. How many 3-PCA we get now? 1. Correct or not? If I have 5 rupees, I want to purchase a chocolate of 3 rupees 1 and 2 rupees 1. How many I'll purchase? I can purchase only 3 rupees is only 1. Correct na? Now because we don't have a 6 carbon to produce 2, we have only 5 carbon. Hence how many we produce? 1 molecule of 3-PCA. How many carbons are left out? 2. Correct na? That is called 2-PGA. 2-phosphoglycolate. Now question is very clear. Is there any use of 2-PGA? Do you think Calvin cycle continue now? Never. If there is no 2 molecule of 3-PCA, Calvin cycle never continue. Now what the plant has to do? Plant has to convert this 2-PCA back to 3-PCA. Then only Calvin cycle will continue. This is a very wasteful reaction. Lot of cell organelles are involved, lot of enzymes are involved, lot of energy is required. That's why photorespiration is a very expensive side reaction, waste reaction. Since what is the stable product here? Sorry, byproduct here. How many carbon? How many carbon it is? 2 carbon. That's why photorespiration widely called as C2 cycle. You're getting the point? So C2 cycle, very good, very good. C2 cycle is only happens in C3 plants. C2 cycle never happen in the C4, never happen in the CAM. Why? Because they are having lot of escaping mechanisms. Where C3 do not have it. What is the reason? Because in the C4 and CAM, principal enzyme is not Rubisco. You are getting the point. Their principal enzyme is PEP carboxylase. I hope everyone got it. Wonderful, wonderful. Very good, very, very good. Now, let's try to understand very clearly. Multiple times asked question, these are all, okay. And blindly believe in your set exams, most of the time you will get the questions from here only, if the question is too tough. Now, what is my goal? Every student focus very correctly, use your brain and try to understand the concept very clearly. There are 3 cell organelles are involved in the photorespiration: chloroplast, peroxisome, mitochondria. Always remember CPM. Now, what my goal? Every one of you focus on the picture. Can you see this? This is RUBP. Correct, na? What it fixed right now? Oxygen. How many 3-PCA I will get? How many? How many 3-PCA I will get? 1. And how many 2-phosphoglycolate I will get? 1. 1 2-phosphoglycolate, which is how many carbon? 2 carbon. So what should I do now? This 2-phosphoglycolate, I have to move to all the organelles. Finally, I have to convert into another 3-PCA. Then only the Calvin cycle will be continued. Otherwise, Calvin cycle will stop immediately. Then plant cannot synthesize its food. How we are going to resolve this issue? Now understand. So 2-PCA, also called as phosphoglycolate. In the chloroplast, listen everyone, it is converted into glycolate. You're getting the point? 2-phosphoglycolate is converted into what? Glycolate. Got it? Now glycolate entered from the chloroplast into peroxisome. This is the question they asked in the exam. Which of the following first intermediate that entered into the peroxisome? First question they asked. Which is the 2-glycolate. Correct, na? Now what happened to the glycolate? Glycolate combined with the H2O2, it is converted into glyoxylate. Where? In the peroxisome only. Later, glyoxylate converted into what? Glycine. You're getting me? Now don't get confused. Now the glycine from peroxisome, where it enters? Wonderful. Now the glycine is getting converted into what? Serine. Now I told to all of you it is an expensive side reaction where we are spending energy unnecessarily. But all of you focus on this. It is reduction reaction. We gain NADH. Understand the point. We gain NADH. So remember this. Then again serine entered into peroxisome. Serine converted into hydroxypyruvate. Hydroxypyruvate converted into glycerate. Now here it is again oxidation. We produced NADH, we spent NADH. So balance. Now the glycerate entered into chloroplast. Now glycerate with the help of ATP, it is converting into 3-phosphoglycerate. Right. This is completely waste reaction. Right. That's why we can call it expensive side reaction. So I'll quickly summarize. Whenever when Rubisco fix the oxygen, we will get 1 molecule of the 3-PCA, then followed by the 1 molecule of 2-PCA. Now that 2-PCA converted into the glycolate, glycolate enters into peroxisome, glycolate converted into glyoxylate, glyoxylate converted into glycine, glycine, again the question asked in the exam, which of the following cell organelle optically inactive amino acid is synthesized in the photorespiration? which is the organelle. The question is, which of the following cell organelle is involved in the production of optically inactive amino acid during photorespiration? Which is the peroxisome. Because what is optically inactive amino acid is glycine. Okay. Answer is same, but the question they twist and ask. So then the glycine convert into serine, serine enter into the peroxisome, serine convert into hydroxypyruvate to glycerate, glycerate entered into the chloroplast, then it is converted into 3PC. Right. So in the case set exam and TS set exam, in both exams they have asked the same type of a question. They have given all these equations or all the reactions in a irregular order. They asked us to arrange them in the ascending order. So you have to remember which reaction comes first, which organelles comes first, which organelles comes second. Okay, that is very important. You have to remember compulsory. Very good, very good. Your time is 10:30. Okay. Okay. Now that is what pathway? C3 pathway. In our exam, 99% they will combine the question of C3, C4 and CAM, then they will raise the question. Hence, it's very important to understand all together. Alright, plant adaptations to prevent the mechanism of photorespiration. So, photorespiration only happens in C3. It never happens in C4 and CAM because they will show lot of adaptations or the escaping mechanisms. How they are going to show? Let's see. Why do we call it as the C4 pathway again? Because the stable or intermediate product that is produced will be the 4 carbon containing compound. As I said to all of you, the plant that grow in a hot and dry climate, only they follows the C4 pathway. Best example I told you, right? All the angiosperms. The standard examples are maize, sugarcane, sorghum and millet. I encourage you to remember little more extra examples, but these 4 are the standard examples they use to ask in the exam. Now, the moment you heard the name of a C4 pathway, one of the common line that they will raise in the exam, C4 plants do not have the Kranz anatomy, which is absolutely wrong statement. C4 plants are always having what anatomy in their leaf? Kranz anatomy. What exactly the Kranz anatomy, how does it look like actually? This is what exactly we can call it as Kranz anatomy. Suppose, suppose if it is a leaf, if you do the cross section of the leaf, you will get the upper epidermis, you will get the lower epidermis. Correct na? You can see that what is these cells? Mesophyll cells. What are these cells? Bundle sheath cells. What's the difference between the mesophyll and bundle sheath? You people can observe, mesophyll cells are always located towards the surface. Correct na? More important, very important, all the mesophyll cells are having lot of intercellular spaces. And you see the bundle sheath cell? They are located very deeper. Can you observe any kind of intercellular spaces between the bundle sheath cell? Not at all. But if the Kranz anatomy wants to occur, it is important both mesophyll and bundle sheath cell both are required. Then how is that possible? How can we connect both of them? We can connect both of them via plasmodesmata, which is called as connecting junction. We read in the cell communication. So that is helps here to connect both mesophyll and bundle sheath. Understand till here? In the tomorrow class we will complete the 90% of the photosynthesis. Okay. Is it clear everyone online? If you have any doubts you can ask. Okay, everyone please join at 10:45. We will continue this particular molecular biology. All right. Okay. Join 10:45, we will continue it. Okay. All right. Thank you all. Thank you.